Hello, and a very warm welcome to everyone joining us for today's Select Science webinar titled From Genes to Discovery, How Next Generation Sequencing Reshapes Cancer Research. My name is Jemima Arnold, and I'll be moderating today's presentation. I'm delighted to be joined by our guest speakers, Dr. Nallasivam Palanisamy, Henry Ford Health, and Zachary Herbert from the Dana-Farber Cancer Institute. This webinar will discuss the rapidly escalating role of next-generation sequencing in revolutionizing cancer research. Following the presentation, we'll have time for a question and answer session. So if you have any questions throughout the webinar, please feel free to submit these in the box to the right of your screen. We will also today have some polls which will be launched throughout the Q&A session. So we invite you to respond to those. Without further delay, I would like to hand over to our speakers, and I would like to thank them both again for presenting for us today.
Hi, hello everyone. This is Nallasivam Palanisamy from Henry Ford Health. First, I would like to thank the organizers for hosting this webinar from Select Science, co-sponsored by Beckman Coulter Life Sciences. So today, although the next-gen sequencing technology is here for a while, but I think it is time to kind of go through the recent advances and what are the future this technology is holding for cancer research. So first, I would like to begin with just giving you in one slide the chronological events that happened over a decade. Starting from 1914, where the first evidence of chromosomal aberration cancer was identified in 1914. Since then, so many technological advancements have happened over a period of time. I think in the 1950s, we identified the exact number of human chromosomes that happened in 1956. Then since then, the chromosome banding technique, the plain staining of chromosomes, we identified as a dark kind of bodies. Then the banding technique came, we were able to see the structural aberrations in the chromosome, that the karyotyping was introduced. Since then, quickly, within like 10 years, there are many chromosomal aberrations, particularly the gene fusions. The Philadelphia chromosome was first identified in 1973 in the chronic myeloid leukemia. Then along with this karyotyping, the PCR and the restriction fragment, red polypopsin, these kind of technologies have developed how to manipulate the DNA. Then quickly in the early 1990s, the comparative genomic hybridization, the fluorescent in situ hybridization, this technology, these are all companion kind of technologies that have developed, have been developed to identify both numerical and the structural aberration and also copy number aberrations. Then quickly in the year early 2000s, particularly in 2007, the next generation sequencing technology was introduced. So since then, this technology is not looking back. So many great advances have come, and we are in a very exciting era. Like we were able to sequence the whole genomes, single cell sequencing, long read sequencing, and we are able to integrate the multi-genomics data to get a complete overview of the cancer genome, which is totally unprecedented, the information that we are gathering. So this slide is to give you the complete overview of what the next-gen sequencing technology can do. So now we can do both whole genome like DNA sequencing. We can do the chip sequencing or methylation and other analysis, looking at the gene regulation functions. Then the transcriptome sequencing, just by sequencing only the RNA, we can do the complete mRNA sequencing. So now the advancement is more on the target capture and the resequencing, like an exome capture, like that. So once you have, even just having the transcriptome or the RNA sequencing alone, one can do so many type of analysis, Like we can look for non-coding RNAs, looking for gene expression, SNP discoveries, novel transcripts, fusion transcripts, and even we can look at the non-coding RNA, because this technology, importantly, is kind of unbiased technology. We can actually analyze the genome in a more unbiased way, like comparing with like microarray and other technologies. Those technologies are heavily biased, meaning that whatever the information that we know, we put it in a microarray, we try to understand what is going on. But the next-gen sequencing is an unbiased technology. You'll be able to sequence everything, whatever is in the sample, both the RNA and the complete DNA sequence, and you will get an unprecedented information. So basically, this technology opened up new avenues for understanding the molecular underpinnings of cancer, accelerating the pace of the discovery, and paving the way for more targeted therapies, particularly in cancer. So with this NGS technology, we can do the improved genetic profiling of any tumor type, and we were able to identify new targets for cancer therapy. And we can do, like by doing the single cell analysis or the liquid biopsy analysis, we can actually efficiently monitor the early detection and also monitor the treatment response. And importantly, the NGS has revolutionized the personalized treatment plan. So, we can thoroughly sequence the samples to identify some new targetable mutations using this technology. And we can understand the drug resistance. And if you look at the advances in the research methodology, it has brought out new methodologies in research, such as single cell sequencing, that have made it possible to study cancer at a single cell level. So now you heard about what is called spatial transcriptomics and other things. We get an unprecedented detail of cancer. So although this technology has made important contribution to cancer research, it may have some limitations, which we will discuss later, but it has made a profound impact on cancer research. Okay, but the limitations, particularly the cost, the data interpretation analysis, is the more challenge. We can generate volumes and volumes of data, but the analysis is challenging because there is no standard way of analysis. This is up to the kind of the brilliance of the bioinformaticians. They can look at the data in all different ways to get out the important information. So with that, using the next-gen sequencing technology, so recently, as you all know, that what is called the T2T, the telomere, the consortium, they are able to sequence the complete sequence of the what is called the 3 billion base pair of the entire human genome. Okay, there is no gap now, because the original version that was published in 2000 was it contained a lot of gaps in the genome, but now we have the complete sequence. And even recently, they were able to completely sequence even the Y chromosome. That was a challenging one that was also kind of completed recently. So that attests the power of this NGS technology. So with that technology, what happened was I think so many agencies have involved, since we are able to actually sequence hundreds and hundreds of cancer samples from at least like 25 or 30 different type of cancer. So all these data, the large volume data, is put together into two kind of major database. One is called the Cancer Genome Atlas. The other one is called the GTEx, the Genome Expression, what is called the Genotype Tissue Expression Portal for Functional Genomics. Okay, these two are the important resources for any cancer researchers. They can quickly refer to what type of genes, whether they are expressed in the normal tissue or in the cancer tissue. We can quickly go through this database. So I can give you just a quick example here. So this is an example, so about 53 different type of normal tissue using the next-gen sequencing data. When you go to the GTEx portal, you can query any gene of your interest. So in this example, I'm showing one example. Since I am actually more involved in prostate cancer research, I'm interested in one gene called the KLKP1, which is a pseudogene. As you can see, this database, just by few mouse clicks, you'll be able to see what is the pattern of expression of this gene. Here I'm showing this particular gene is expressed only in prostate, not in any other kind of normal kind of human tissue or organ. So this this is the powerful kind of information that you can quickly generate. Then all you have to do is we are trying to understand what are the functional consequences of this gene like that. So this is by using the GTEx portal and the TCGA. Again, there are many kind of open source databases like the CBio portal that is quite popular. They have curated all the cancer genome atlas data of all, I think about 80 or 90 different type of cancer data are actually curated, and they are ready for any viewers. Just with a few mouse clicks, you will be able to understand what is going on. So here in this example, I'm just showing the PIK3CA mutation across 89 different type of cancer. What is the frequency of this mutation in the across you can look at a pan-cancer view of any gene of your interest, and based on this, you will be able to generate some hypotheses, then go for validation and try to understand this thing. So this clearly tells the power of this next-gen sequencing technology, both looking at the normal genome and also on the cancer genome. So here, what I'm in the rest of the presentation, I'm going to highlight, since I'm actually more focused on prostate cancer research, I'm going to show how this technology, the NGS technology, has really helped to advance prostate cancer research. So I'm more particularly interested in looking at the gene fusions in solid cancer. So we employed this next-gen sequencing technology in our research. So here I'm going to show you how the important discoveries were made before the next-gen sequencing technology. So before the NGS, one has to engage in like out-of-the-box thinking. We have to come up with some novel approaches to make some important discoveries. So here I'm highlighting some few examples, like by using the cancer outlier profile analysis of the entire gene expression microarray data, like the Affymetrix or other data. So they were able to kind of pull together multiple data sets and try to understand what is the particular, any particular gene that is always stands out, or an outlier gene that is expressed. So using that information, the Eds family gene rearrangements was discovered in prostate cancer. But in the lung cancer, using the retroviral cDNA library, the ALK gene fusion was discovered. Then by using the array CGH, we reported the first ever recurrent gene fusion in cancer, particularly in gastric cancer. So these are, one has to come up with some new ideas, how you can make discoveries. But the next-gen sequencing technology has changed everything. You don't have to think anymore. You don't have to look for any technologies. As I mentioned, since it is an unbiased technology, just all you have to do is, you have to identify the important sample sets, just put together for next-gen sequencing, and you will be able to identify some important biomarkers. So here, as you can see, with the introduction of the next-gen sequencing technology, there are many recurrent kind of gene fusions have been identified in the rare subtypes of different type of solid cancer, like even like liver cancer. We did not identify anything before, but with the next-gen sequencing, and also even in the hepatocellular carcinoma, there are new kind of gene fusions have been identified. So this is just to show you the power of this technology, how it actually helped to identify some new information in cancer. So these are some of our publications. So we adopted this technology very early when it was introduced, because I wet my hands making next-gen sequencing libraries in 2007 itself, even before the introduction of the illuminate called Solexa. That was the early version of the next-gen sequencing. That time it was only single read sequencing, not even paired end sequencing. So we were able to make some important discoveries, particularly in prostate cancer. We looked at both the protein coding gene landscape, and also we looked at the non-coding RNA area as well. So here I'm going to just give you one example how we made a very important discovery in prostate cancer. Just by sequencing I think about 25 samples using next-gen sequencing, we were able to identify just only one sample that was showing very high or outlier expression of the gene called the BRAF. As you all know, this is a kinase, it's a tyrosine kinase, it's a druggable important target. And it is heavily mutated in melanoma and other cancer. And this is the first time by using the next-gen sequencing technology, we showed that the overexpression, so just all we see was it is a very highly expressed. But when we went deeper to characterize the expression pattern, what we did a kind of PCR by using exon spanning primers. So we identified like only from exon 1 to 8, there was no expression at all. Then after from exon 8 and the rest of the sequence are highly expressed. So which clearly showed that, this is a fusion gene because the full-length transcript was not completely expressed. So we did characterization, then we identified the fusion pattern is the SLC45A3, which actually fused with the BRAF. So this is the first time, we discovered recurrent druggable gene fusion in prostate cancer using next-gen sequencing technology. Otherwise, it would have not been possible for any other methods that we have. So then, since then, the Cancer Genome Atlas, what they did, they sequenced more than about 333 prostate cancer samples, including Caucasians, African American, everything. They were able to characterize, or in other words, we can actually classify the prostate cancer into distinct molecular subtypes. There are at least like 7 different kind of recurrent genetic aberrations have been identified. And now we have first ever molecular classification of the prostate cancer. So, so using this, when you come to the classification, these are the important kind of gene fusion that are identified across prostate cancer, like the S gene fusions, ERG, ETV1, ETV4, ETV5. I was involved in the discovery of the RAF kinase gene fusion. So now using the next, since I told you this is an unbiased technology, we were able to look at the non-coding RNA landscape as well. So I will show you in a couple of slides what we discovered in the non-coding landscape. So what is the highlight of this this slide is that we were able to classify the prostate cancer into distinct molecular subtypes, because we did not identify a single aberration that is present across all the patients. Each aberration is identifying only a small subset of prostate cancer. And interestingly, those kind of gene fusions have some potential therapeutic possibilities. There are many researches going on to develop targeted therapy for the gene fusion. So all these are made possible with the introduction of the next-gen sequencing technology. So now quickly about not only the protein coding gene landscape, the non-coding RNAs are also plays an important role, because we thought most of the, except like 1 or 1.5% of the human sequence is coding for proteins, and the rest we thought these are all junk DNA. But that idea is completely kind of changed because of the next-gen sequencing technology, because now we say that more than 99% of the human genome is transcriptionally active, where the non-coding RNAs are playing a very important role in the regulation of genes and other things. So I will show you quickly what we did in this non-coding RNA landscape. So by using the next-gen sequencing technology of 13 different types of solid cancer, what we did was we looked at the expression pattern of pseudogenes, because pseudogenes are the another class of non-coding RNA. So these are not the typical long non-coding RNA. These are all the defunct genes of the wild type, the genes that are already in the genome. So what we did using the next-gen sequencing technology, we were able to identify tissue-specific and also cancer-specific expression of pseudogenes across all these 13 different types of cancer. We made some important discovery using that. So just to give you an example, for those who are interested in looking at the pseudogenes, as you can see, for a single gene, that is a normal protein coding gene, there could be like several pseudogenes you can identify. Like for this particular gene RAN, there are about 15 pseudogenes. They are located in different locations in the genomes have been identified. Like that, many genes carry, like a different pseudogenes. So until the next-gen sequencing, we all thought that these pseudogenes are like dead genes or they are not functional. But based on the next-gen sequencing technology, the first time we showed that these pseudogenes are indeed transcriptionally active, and they are highly expressed more in a tissue and a cancer-specific manner, and they have some important regulatory functions. So interestingly, what we did was, when we did the pseudogene analysis, since I was actually more interested in looking for gene fusions, we identified a very interesting gene fusion in prostate cancer. So what is happening here is a pseudogene called KLKP1 that is located on chromosome 19 is actually fused with a protein coding gene called KLK4. So the first two exons of the KLK4 fused with the exon 4 and 5 of the KLKP1. So by nature, the KLKP1, the pseudogene, are kind of non-coding genes. They don't make any proteins. What happened was, in this example, after the fusion, because of the shift in the open reading frame, what happened was the part of the KLKP1 pseudogene is converted into a protein coding gene. So there are about 55 new amino acids that are completely coming from the pseudogene. So this is a first ever rare event where we showed that the involvement of a non-coding pseudogene in the formation of gene fusion in any solid cancer. And it is very rare, and it's important to note that the KLKP1 pseudogene, now it is converted into a protein coding gene. And when we looked at the expression of this fusion gene across different cancers, we found it is expressed only in prostate cancer, not in any other cancer, which shows the specificity of this. So like I showed you before, using the GTEx portal, the KLKP1 gene that is expressed only in normal prostate. But what we see that the fusion is expressed only in the prostate cancer. So this clearly shows, the importance of this gene in prostate cancer, and also we are able to identify the gene fusion only because of this next-gen sequencing technology. So, so this is again, I want to show you the same slide that the KLKP1 is very specifically expressed only in prostate. This is the normal full-length KLKP1 that expressed only in the normal prostate. You will see the fusion in the cancer. So like that, using next-gen sequencing technologies, not only in prostate cancer, all other kind of solid cancer have also been kind of extensively investigated. There are many multiple genetic mutations have been identified in all different type of cancer, particularly lung cancer. So that leads to the what is called the personalized medicine. So now we were able to classify the patient into distinct molecular subtypes. So just by sequencing the individual samples, you can identify. So suppose in a cancer, if you have multiple mutations, you will be able to identify which particular mutation is present or absent in a particular samples. And if there are targeted drugs or therapies available, the patients will get benefit on that. So that is the power of the NGS technology. So the next important event with the next-gen sequencing technology is the understanding the tumor heterogeneity. So any solid cancer, it is very rare to identify, like a homogeneous pattern of the tumor. So even within the tumor, there will be multiple kind of variations, what is called the intra-tumor heterogeneity. So next-gen sequencing technologies really helped to understand this tumor heterogeneity as well. Like in this slide, there are different areas. What you can do is, you can sequence different areas within the tumor, and you can actually identify, a lot of structural variations, copy number changes, mutations, even within the tumor in a different location. So I don't have much time to go into the details, but just to show the importance how, the next-gen sequencing technology can help to understand the tumor heterogeneity. So this is again, so when you have, this is the prostate, the entire prostate, it is well known for multifocal cancer, meaning that at least 3 or 4 tumors will be there in each prostate. cancer patients. And what we showed that based on our analysis, we clearly showed that the different tumor foci carrying different driver molecular operations. So that is the information we are able to get using the next-gen sequencing technology. So again, to show the importance of the tumor heterogeneity here. So this is a patient with the BRAF mutation melanoma who had cancer all over his body. And there was an important drug that was developed called the PLX4032 targeting this mutant BRAF. And after giving this treatment, the cancer was completely cured in this patient. But after like 15 weeks of therapy, the tumors started coming back. Meaning that what is happening is only the tumor cells carrying the BRAF mutation responded to the drug, and the other cells that do not carry the mutations, they started growing back. So that clearly shows the tumor heterogeneity. So we were able to, then if you have in this kind of situation, what we can do, we can take some repeat biopsy samples after the relapse, then subject them for next-gen sequencing, more in-depth sequencing. You may be able to identify some new kind of mutation that may be targetable that will help the patient. So that clearly shows the power of the NGS to understand this tumor heterogeneity. And using that technology, even in prostate, we showed that by the existence of multiple kind of molecular subtypes carrying like one or more than one kind of mutations in the prostate cancer. So these are all completely new information, and we showed that these different kind of molecular subsets have some implications in the outcome in terms of recurrence-free survival and other things. It has an importance. So these are all made possible only because of the next-gen sequencing technology. So what is next? Right. So we have come a long way using this technology. So we need to actually focus on the marker negative. So we did identify several molecular subtypes of prostate cancer. Still on the top, the first row of this table, as you can see, still there is another subset where we don't know what is the genetic mutation going on there, because these subtypes of cancer are completely negative for all the known markers. So these are the important subset of the prostate that we are focusing on, try to understand or try to discover some new molecular markers. So for that, so particularly as you all know, in the prostate, it's very difficult to collect some fresh frozen samples, but we have a large kind of collection of FFPE tissue. So we have to come up with some new technology and approaches. I'm sure I think the next speaker is going to highlight how to improve the analysis of the FFPE tissue. But here I will touch upon that how we did. So what we did, we carefully selected these kind of biomarker negative samples. Then using the FFPE tissue, we were able to isolate the DNA and RNA. We subject them for high throughput and next-gen sequencing analysis by using some important, because the RNA isolation is a challenging one. There are, although different methods are available, but we are using the Beckman-Coulter, the PharmaPure XL RNA. We did some kind of pilot study, and we got some interesting results using the RNA. We subjected to next-gen sequencing, and we made some important discovery. So this clearly, particularly the African American samples we used, and we compared the fresh frozen RNA versus the FFPE RNA, and we are able to show some concordance because of our ability to isolate some good quality RNA using this new RNA isolation methods provided by the Beckman-Coulter technology. So we using this approach, we were able to make some important discovery. In fact, we identified a new recurrent gene fusion, particularly in African American prostate cancer, which we are in the process of kind of characterizing these gene fusions. So in summary, what I would like to highlight is, the NGS really created a revolutionary impact on cancer research. Because of the unbiased nature of the NGS approach, we are able to make some important discoveries. It really overcomes the limitations of traditional methods like karyotyping or Sanger sequencing, and the high throughput approach really created a transformative impact. And like I highlighted, extensive application in precision medicine and enabled identification of unique genetic mutations leading to personalized treatments. and the fueled large-scale projects like the TCGA and the GTEx. And now the era is whole genome sequencing for comprehensive mapping of all genetic changes in the cancer genome. And it helped to identify some new prognostic and diagnostic biomarkers and drug targets. So overall, the NGS really accelerated cancer research from gene-level insights into practical applications in the diagnosis and treatment. So I would like to acknowledge, because it's a large team effort, we have been working on this cancer cell more than like last 20 years. So I want to thank everyone. So with that, I would like to hand over this presentation to the next speaker, Dr. Zachary. Thank you.
All right, thank you. I'm Zach Herbert from the Molecular Biology Core Facilities at Dana-Farber Cancer Institute. And I'm going to tell you about how we've been using automation to solve some of the cancer-specific problems we have related to difficult samples, and then also working with difficult methods or methods that are complex and long, and all mostly related to cancer research. So first, just to tell you a little bit about our core facility and our institute. So the Molecular Biology Core Facility, or MBCF, was the first core facility at Dana-Farber, founded in 1985. And our main mission is to provide access to cutting-edge instrumentation technology, and then provide associated expertise for the investigators at our institute to really get the most out of these new technologies. We support basic and translational research, which is another way of saying we are not a clinical diagnostic lab. And most of our services are related to genomic and proteomic applications, although recently it's been pretty heavily weighted towards genomic applications. One of the main reasons is next generation sequencing is one technology that's good for many different things. Here's just a slide full of different acronym seeks methods that we have firsthand experience with. It's a very small subset of the methods that are out there, and even probably the methods that we've worked with. So there's a lot of different and powerful approaches that can be used to explore biology using this next generation sequencing. For the more common methods, such as RNA-seq, for example, that's one of the highest volume services that we provide. We extend beyond just the sequencing. We need to get the samples ready to go into the sequencer, and then when the data comes out, instead of reporting just really large files full of 4 letters, we like to do a little bit of work to get our investigators a little bit closer to interpretation and biology. So we also build bioinformatic pipelines on the back end. And then even within one acronym seek, so this one's RNA-seq, there's still at least 5 or 6 different methods that can be used, or more, to handle that type of sequencing. And this is just kind of a decision tree that we use to kind of navigate, based on sample quality and quantity, what the most appropriate method is. Oh yeah, and also the biological goal too. But unfortunately, especially in the cancer world where we're working with difficult samples, a lot of times it's the sample itself that determines the most appropriate method. I'll come back to this a little bit later. And here are some other examples of these kind of end-to-end full service workflows that we've developed somewhat recently, including low-pass whole genome sequencing to do copy number analysis, ChIP-seq, and its more recent relative cut and run, exome sequencing, and amplicon sequencing. So these are all things that we do, library prep, sequencing, and provide some back-end analysis as well. And how we do this is kind of a 3-pronged approach. So our core is primarily built out around different instrumentation, sequencing technologies, and as well as automation. And then we also have to we're constantly testing and evaluating new sample preparation methods to go into this instrumentation, and then, as I mentioned, building out bioinformatic pipelines on the back end to really try and leverage the most out of this. And it's really important as well for optimizing and developing these pipelines that we have a way to know how we're doing in that evaluation and optimization process. So I'm going to tell you two stories, recent stories that are kind of relevant and highlight everything I just described that we do in our facility. So DNA methylation is an important modification to the DNA, and it has implications for gene regulation, both in normal and pathological biology. And so being able to profile the methylation status across the whole genome is a powerful tool, both to just characterize normal biology as well as cancer, and potentially have some prognostic and diagnostic applications as well. We had this project come up with where they wanted to do this whole genome profiling for multiple myeloma, and had blood samples from over 600 patients. And we at the time were interested in this new approach to methylation sequencing called enzymatic methylation. It was developed by New England Biolabs. The classical way of doing methylation sequencing involves using sodium bisulfite, which is a chemical conversion that converts all the unmethylated cytosines into thymines. And so the sequencing readout, any place you see where there is a cytosine that's preserved, was originally methylated. So you do this chemical conversion, and then you're basically flipping all of the unprotected C's to T's, and anywhere in your readout where you see a C, you know it was originally methylated. Enzymatic methyl-seq works the same way in that you put the same DNA, genomic DNA, into the reaction library prep, and the readout is the same, but how you get there is a bit different. Instead of using a chemical treatment, there's a series of enzymatic reactions that give you the same kind of conversion output. And the main benefit of this is the chemical conversion is kind of harsh, so it can cause excessive degradation in DNA. And if you're working with cancer, potentially DNA is already highly degraded. And then you get pretty uneven distribution of your coverage across the genome, which is another way of saying areas that you might be interested in might not be covered very well, and then areas that you're not interested in might be covered kind of excessively. So one of the nice features of the EM-seq approach, which is also indicated in this figure on the right, is that you get more even coverage across the entire genome, and especially in areas of methylation. So the green lines are showing at the same coverage, you're getting more, the same depth of coverage, you're getting more CpG regions covered than you would with whole genome bisulfite sequencing. So the kind of the original method at the same level of sequencing across, and this is also showing across different input amounts. However, there is a bit of a trade-off. The EM-seq method is, because it involves these two conversions, is a little bit longer and more laborious to perform in the laboratory. So in our group, we have two Biomech i7 automated liquid handling stations that we use to do all of our high volume library prep for next generation sequencing. And there's some nice features of this i7 platform, or it has a large deck space. This is actually our instrument in the lab, and I've kind of blown this up here so you can see the different positions. So it has a large deck, which means you can put lots of reagents and consumables on the deck, which allows you to work with more complicated protocols without having to have lots of manual interventions. We're also fortunate enough to have automated thermal cyclers on the deck, which makes our methods even more walk away. And it's a great solution for handling approaches like EM-seq that can be challenging to get working manually in the lab, and not just that, across for hundreds of samples. Processing hundreds of samples manually using a complicated protocol is not feasible. In this case, we collaborated with Beckman Coulter and NEB to automate EM-seq on our i7. It was a new method, so it hadn't quite had a chance to for the automation protocol to be developed. So we worked to get it developed. And as I mentioned before, in tandem, in order to know how we're doing and is the method working and what kind of tweaks do we need to make, we also developed a QC pipeline, specifically Lutz and Michael from the computational group that supports our laboratory. This was really helpful, both in terms of getting the method up and running, but also when we're into projects now, we have a way of, we can either run this on the full data set or take a light subsampling of the data and run it really quick through to see how the conversion was. That's the main kind of metric that we kind of base our primary QC on. And we do that by spiking in 2 control DNAs into each sample. One of them is this lambda DNA that is synthetically totally, actually it's not synthetic, but it's totally unmethylated. So we don't want to see, we wouldn't want to see any cytosines in these samples. And then we have this PUC19 control spike in that should be close to 100% methylated. So our pipeline basically looks at these 2 control spike-ins. So you can see we want to see the green bar close to 100%, and we want to see the blue one down close to 0%. And then we're also mapping to the human genome in this case for that purpose. So back to the multiple myeloma project. After automating the workflow, getting it optimized, we were able to prepare and sequence samples for 650 patients. And analyzing 650 whole human genomes is not a trivial task, so they're still working on that analysis. But because of our pipeline, we were able to QC all of the data to confirm and that we achieved good conversion rates. So at least from a technical standpoint, the methylation data for this project was high quality. So now I'm going to shift gears a little bit and talk about how we would handle, or how we handle challenging samples. So as Dr. Nalla mentioned, FFPE is formalin-fixed paraffin embedded tissue. There's a lot of these samples. They've been archived for decades now with complete clinical outcome and treatment records. So it's super powerful for retrospective studies, but they're notoriously difficult to work with. Specifically, RNA, you often get low yields. Quality metrics are not good. There's lots of factors that go into contributing to this, such as the extraction method, and even like the protocol for the fixation and embedding itself, and then the storage conditions. So I just want to take 1 second. So the most important metric, RNA quality metric that we rely really heavily on in our laboratory is the DV200 score, which is the distribution value greater than 200 nucleotides. Basically, this is the fraction of your sample that's viable to be converted into a sequencing library. So we want to have RNA that's greater than 200 nucleotides. The classical metric that's used for RNA is the RIN score. I think it was developed by Agilent when they came out with the bioanalyzer. It's a score from 1 to 10. 10 is like beautiful, high quality RNA, and 1 is highly degraded RNA. The problem is it's mostly a binary indicator. If it's better than 7, it's very good. If it's less than 7, it's maybe not, but it doesn't really give you a good sense of how degraded the RNA is or not. And I have an example here over here. So if you look, the RIN scores for these 2 samples are basically the same. This top one has a DV200 of 12%, and this bottom one has a DV200 of 60%, which is another way of saying we can probably generate informative, useful data from this one, and it's probably not even worth proceeding with this this top sample, which is not information that you would get from the RIN score. To make matters worse, even within one project, if we do multiple extractions, we may see a broad range of different outcomes. And again, this is related to all those other factors. So here's a figure from this paper in 2007 where they tested different storage conditions. So they took 3 different tissue types, extracted the RNA, and evaluated it at time 0, and then stored it for a year at 3 different temperatures. So at 4 degrees, at room temp, and body temp. And as you can see, the temperature that the tissues are stored in, it makes a big difference. If you keep it cool, you can still see, even after a year, there's pretty well-defined ribosomal RNA peaks. If you're not used to looking at these things, these are kind of these are good-looking samples that we wouldn't be too afraid to work with. And then on the other end, these are highly degraded and potentially be quite problematic. Extraction method really matters as well. I'm not going to go through this in great detail, but just to say there's lots of different sample prep or extraction protocols that are available to use. And you get some pretty wide variability across protocol and then across different sample types. So this is another challenge working with these FFPE samples. So I'm going to finish up by telling you a very recent story about this endometriosis project, which I understand is not exactly cancer, but the samples were all from formalin-fixed paraffin-embedded tissue. And we were hoping to get around 600 samples that were able to sequence. And knowing the troubles with FFPE, the group actually submitted almost 1,000 samples to us, expecting a large number to not meet quality requirements. And the plan was to do total RNA sequencing using the ribosomal RNA depletion method, which is compatible with degraded RNA. So the specifications for that protocol are we need at least 50 nanograms, and we want to see a DV200 that's greater than 40%. Unfortunately, after our first initial QC, about of those 900 samples, maybe 15% of them met this requirement, which is a problem. So now what? Well, back to this RNA-seq decision tree. If you kick around all the way down to the bottom, you get to this hybrid method where we do an RNA library prep without any enrichment or depletion up front, and then we do a hybrid capture similar to what would be used to do like a DNA exome, for example. And so it's a targeted RNA sequencing approach. Here are some examples of when this might be useful. So it's not just for horrible FFPE samples, but if you were at a specific transcript or set of transcripts that you were very interested in, this might be a viable approach. Or if you're looking to do some functional variant calling and you want to boost the sequence coverage for specific genes, or you're trying to look for something that's very rare and you want to pull that out of the transcriptome without having to sequence a billion reads. And it also happens to work well with smaller input amounts from degraded tissues such as FFPE. So that's what we were going to use. And here's a cartoon of the hybrid capture protocol. So as you can see, after you've made the library, there's still a bit of processing to do in order with these biotinylated probes that are then captured using streptavidin beads. But now we're in a situation where we've got 2 full kind of NGS type protocols that we need to use in order to handle these hundreds of samples. And so this is screaming, please automate me. I think technicians would be quitting and there'd be major batch effects if we tried to do this manually. And in this case, we actually had both of these methods. We already have them validated and installed and run regularly for different applications. We were able to put them together to make to make this project happen. And some other nice features with the i7 are that it gives you these nice deck layouts again, so you can set things up. And if you're making master mixes, it provides these little cheat codes, so you can kind of so almost anyone with a minimal amount of training can operate and run these protocols through the user interface. So back to the project, because we had these 900 samples, even within there was pretty wide variance in the amount of RNA and the quality of the RNA. So we took samples kind of spanning all the different kind of input amounts and qualities that we observed. And that's represented by all these different colors. It's maybe small and you can't read it, don't worry. The point is that across all the samples, we were able to detect over 10,000 genes, which is great. And we had good coverage for the protein coding genes. That's what we would expect. And a couple of these samples you'll see that didn't perform well, they were actually from an original pilot from like 2 years previous. And we only had a couple microliters left, and we just threw them in there to see, like, oh, I wonder how they'll do. And they didn't do well. Some of them did, and it was still ultimately a pretty encouraging result. So we were able to move ahead and just recently finished sequencing all the libraries we could, which is a total of 642. And we were able to do this with low inputs and highly degraded RNA, and it would not have been possible without automation. So just to summarize the stories, leveraging automation has been really important for our lab, both to support samples with high volumes, but also to work with complicated protocols that are beyond the scale of what a human can do very well. And these are the people that do all the work in the group. So I've got the MBCF staff here, the informatics folks that help build our pipelines and support data analysis. I'd like to thank Beckman Coulter Life Sciences for inviting me to speak. And that will do it for me. Thank you.
Okay, thank you for that very informative presentation, both. It is now time for our question and so if you have any further questions, please do send those in. You can submit these by typing into the questions box to the right of your screen. So the first question I've got is actually for you, Zach, and it's what benefits did you find using the Biomech i7 compared to other automation instruments?
Yeah, so there's a number of benefits that we particularly like about the Beckman i7. One is the user interface is it's pretty intuitive and easy to use. You do not have to know how to code or program in order to do it. So we've been able to train most of our technicians to operate the instrument. And without, there's not like a big barrier to entry in order to operate it, which is great, and not the case with some other automated liquid handlers. The methods that we get from Beckman, where we don't have to collaborate to develop them ourselves, have already been fully wet lab tested and validated, both in their internal group and then external. So it's and we have a standardized deck layout. So both the decks on both of our sevens are the same, and it's also similar to the standard NGS configuration. So methods are easily transferable, and usually within a week of installing a validated method, it's up and running in our group. So that's great. And there's some other, there's a 96 head that's great for stamping reagents, and then there's a span 8 that has the ability to get in and out of different sized labware and do more refined pipetting. And then there's also 2 grippers that can pick up plates and move things around. So it's an efficient system as well.
That's great. Thank you for providing that for us. So I think I've got a question here for you now, Nalla, and it's why did you choose former pure chemistry to extract RNA? I think you're on mute again there.
Yeah.
That's lovely.
So yeah, I expect this question. So we use pure chemistry, especially for RNA extraction. It is primarily because of its high compatibility with FFPE samples, because that is the only source of tissue that we have available for our studies. So I think the pure technology ensures high quality and a high yield RNA, which is essential for our downstream applications like RNA sequencing. And also it integrates seamlessly into automated platforms. Suppose if you have any method for extracting RNA, and it reduces the potential for manual errors and increasing the efficiency of RNA isolation. I think more than that, its versatility, allows us to conduct multiple types of molecular analysis, such as DNA and protein studies from a single sample. So this is particularly useful for comprehensive studies like ours, that particularly we are interested in looking at the tumor heterogeneity and other things. So I think, the pure is an industry standard method, which I would attest, which lends credibility to our research methodology. So I think overall it aligns well with our project's objective and the resources that we have at our hand. Thank you for the question.
Okay, that's lovely. Thank you, Nala. And then I think this next question is a bit more geared towards your presentation, but obviously, Zach, if you have any expertise to offer, please do. But it's asking, how is NGS being used to understand cancer heterogeneity?
Yeah, so like I highlighted in my presentation, so NGS has been instrumental in decoding cancer heterogeneity, because I don't think any other method you can actually understand at this level. So the genetic and the epigenetic variation within and among tumors that you can study using NGS. It also allows the multi-regional sequencing, like I pointed out, because of the extensive tumor heterogeneity. Even if you sample different sections within a tumor, you are going to see so many variabilities. And it also allows researchers to examine the epigenetic factors like DNA methylation and histone modifications by using the ChIP-seq and other methods. And I think the NGS can be applied to study transcriptomic variability through RNA-seq at a single cell level to provide more granular understanding of cancer diversity. I think importantly, the repeated sequencing over time or in response to treatment can track how a tumor evolves. So these are all the information that you can gain only by doing this unbiased NGS sequencing, particularly the liquid biopsies. It can offer a minimally invasive approach to this type of monitoring. So I think these comprehensive insights from NGS are increasingly used to tailor the personalized therapies and understand resistant mechanisms, and thereby it is revolutionizing cancer treatment. Thank you.
That's lovely. Thank you. Zach, do you have any input for that one? Or I can move on to another question.
I would just say, in addition to the single cell approaches, kind of spatial resolution for either at single cell or very small cellular subpopulations is kind of like the cutting edge of NGS right now, that really gets at tumor heterogeneity, and not just the heterogeneity, but where it is specifically in space.
Yeah.
Okay, lovely. Thank you. And then I think our final question for the day, just in our final few minutes, is for you, Zach, and it's what benefits are you expecting the Echo 525 to bring to our process?
Oh yes, so the Echo. I didn't have quite enough time to talk about that as much as I'd like. So the Echo 525 is a liquid handling instrument that's super cool because it uses sound waves to move 25 nanoliter droplets from a source plate to a collection plate, which turns out is super powerful. We're using it actively right now to normalize and pool large volumes of NGS libraries, so that when we put them on the sequencer, we get a perfectly balanced read distribution. It's a great application. It's not the most exciting. The other more exciting thing that we're using it to work on now is miniaturizing sample prep reactions, which is another way of saying reducing the cost and the sample usage for our library preps, so that for the same dollar you can get more, you can process more samples, which is obviously of great benefit to these larger and smaller projects that we're handling for cancer research.
That's great. Thank you very much, both. That is all we have time for today. So I would like to thank our expert speakers once again for the informative presentation and discussion. And a very big thank you to everyone joining us online. We hope you have found this a worthwhile session. If you have any further questions, please feel free to email me at editor@selectscience.net, and I will follow up with your questions for our speaker. Remember, you can download related resources in the tab to the left of your screen, and this includes the certificate of attendance. If you'd like to listen again to today's webinar or invite a friend to listen, it will be available to watch on demand in just a few days. Goodbye, and thank you once again for joining us.